Hello class, this is part 5 of organic molecules. It'll be over nucleic acids. Alright, so the building blocks for nucleic acids are called nucleotides. So nucleotides are the monomers, the building blocks. The polymers are called nucleic acids. There are two types of nucleic acids, DNA, which is deoxyribonucleic acid, and RNA, ribonucleic acid. But always keep in mind they have the Na in there for nucleic acid. You need to know your functions of each. DNA is for information storage. RNA basically makes proteins. So DNA stores the information to make those proteins, and RNA actually makes them. Your nucleotide is made up of 3 parts: a phosphate group, which if you remember is a PO4, and it's at least a 2-, it might be 3-. You have a pentose sugar. Pentose just means it's 5 carbons, it's 5-sided. It'll either be ribose for RNA or deoxyribose for DNA. And then we have a nitrogen containing base. There's lots of nitrogen in it. So each amino acid had 1 nitrogen. These bases have a few. And you need to know the names of the bases, so adenine, guanine, cytosine, and thymine are the ones present in DNA. In RNA, you don't have thymine, instead you have uracil. So if you're looking at a strand of RNA, you could tell it was RNA because there were U's and not T's. Similarly, you could look at a strand of DNA and tell it's DNA because there's thymine and not uracil. So here's your nucleotide. Here's your pentose sugar. Here's your nitrogen base, nitrogenous base. And then they're representing the phosphate just like a P, but it really looks like this. Alright, so deoxy, D means to remove or away, and oxy is talking about oxygen. So the difference between deoxyribose and ribose is this oxygen right here. So for now, just know that deoxyribose is missing an oxygen. Later, when we get to the DNA chapters, you may actually have to recognize it. But also recognize that these are sugars, they're carbohydrates, because they have that ring shape, even though it's 5-sided, and lots of OHs. Here's your nitrogenous bases, and this one has 3 nitrogens, this one has 2. They kind of vary between 2 and 3. These you don't need to know, well, maybe you will. So purines are going to be adenine and guanine. They have 2 rings that are fused together. 1 ring there, 1 ring there. And remember, fused means there's no space between them. They share this wall. And then your pyrimidines are just 1 ring. And that's kind of how I remember it. The pyrimidines with the longer name are smaller. They have one ring, and they're cytosine, thymine, and uracil. The purines with the smaller name are the larger structured, and those are adenine and guanine. Okay. So the last thing I want you to know about the bases is they hydrogen bond, they're complementary. And cytosine will always hydrogen bond with guanine. And adenine will hydrogen bond with thymine in DNA, but it will hydrogen bond with uracil in RNA. Let's just look at it. Alright, so here's the basic structure of a nucleic acid. You get the covalent bonds between the sugar and the phosphate. And so this is how they're linked together. And the bases will kind of hang off to the side. This is called the sugar phosphate backbone, this ribbon, where it's alternating phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, and that can go on forever, Some of these nucleic acids are really long, especially DNA, and it's amazing that they fit in not only in a cell but in a nucleus, which is also due to folding. If you look at DNA, it is okay. So RNA was single-stranded. DNA is double-stranded, and this should give you some hints on its permanence, on its stability. So double-stranded nucleic acid is more stable. It's better for storage than single-stranded DNA, which can be attacked by enzymes and destroyed. When I worked in a lab, anytime we were working with DNA, we would just work at our bench. We'd wear gloves, we'd store stuff at -20 or refrigeration temperature, 4 degrees Celsius. But no big deal otherwise. But anytime one of my lab mates was working with RNA, they would clean their desk, they'd put a sterile pad down, gloves, goggles, face mask, everything, just because RNA gets destroyed easily. And so if you're working with it, you want to make sure it's not destroyed so you can detect it in your experiments. So DNA is double-stranded, it's more stable. These blue ribbons here are the sugar phosphate backbone. In the middle here you can see the bases hydrogen bonding, and that's why I was talking about the complementary base pairing. So since this is DNA, they're going to show adenine with thymine and cytosine with guanine. The cytosine-guanine pair make 3 hydrogen bonds. The adenine-thymine pair only make 2. So you can see effects of that when we start getting into biotechnology. You can you can separate the 2 strands of DNA easier if there's lots of A and T's in there, and that's because they have fewer hydrogen bonds, just 2. Alright, so a modified nucleotide is used as our main energy molecule in the cell, and that modified nucleotide is adenosine triphosphate. Tri just means 3. And so ATP has 3 phosphates. And it'll be used in the cell to do work. Again, it's the main energy molecule in the cell. The reason our cells eat, the reason they eat glucose, is to make ATP. So if we look at it, here's adenosine. So we're used to nucleotides being a nitrogenous base, a sugar, and a phosphate. So the base in ATP is adenine. The sugar, I believe, is ribose, and then it has 3 phosphates linked together. Now, if you draw these out, I kind of hate that they're simple P's. There's an O there, then a P. I'm gonna run out of room. Let me draw it where I have more room. So the phosphate will be connected to your ribose via an oxygen. And then it'll sort of alternate. And there's the third. So the main reason I drew all this out and spent like a minute doing it is to show you there's a lot of negative charge in this triphosphate. And remember, in chemistry and physics, opposite charges attract. Well, also same charges repel. So all these negative charges don't want to be near each other. And that means there's a lot of energy in these bonds. So it's why ATP has so much energy and why it's the energy molecule. Now typically we only break this last bond, so we end up with ADP, which is diphosphate. Di stands for 2. Or triphosphate stands for 3. And then we'll end up with a free inorganic phosphate, and we'll release lots of energy. There is some energy in that second bond, and then you get AMP. The M stands for mono, which is 1. And then you'll get 2 phosphates by itself, which we call inorganic because they're not connected to carbon anymore. Over here, they're connected to carbon. The sugar and the adenine both have carbon. So once they're free from the structure, the adenosine, they're no longer attached to carbon, so we call them inorganic phosphates. Anyway, So anytime we're breaking off a phosphate, we get more energy, although there's less phosphate in that second bond than in the first. And when we make ATP, we typically add these two together. And we use energy that we freed up from glucose to sort of staple them together to reform that bond. So for nucleic acids, know the structure of a nucleotide. Know that nucleotides are the monomers, the building blocks, and nucleic acids are the polymers. Be able to compare RNA with DNA. So RNA is single-stranded, DNA is double-stranded, RNA is ribose, DNA is deoxyribose, RNA has uracil, DNA is thymine. Some other things I may test you on. RNA can be anywhere in the cell, but DNA is typically restricted to the nucleus. The only time DNA is let out is during cell division. And then function. Know that DNA is for information storage, storing genes, and RNA is for making proteins. And then know the structure of ATP and know that it's the main energy molecule of the cell.